Take the moment to address and introduce you to Andrea Souza. Andrea and I have actually worked together in previous lives, so I've known her for a number of years. She's a dear friend, so I'm really very excited to hear her talk, and I'm going to think up some really tough questions for her.
I think Barb's going to get that all covered. So I think the best part and the worst part of being at this point in the day is we're all really tired, and the only thing that stands between us and the power walk is a couple more of these talks. So hopefully I will do the subject justice. I just wanted to follow up to a comment about ChemAxon. At least my perspective, I met Alex, I think 12 years ago, a little after I met Doug, was that ChemAxon kind of stood for chemistry and biology coming together because the axon is the part of the nervous system that conducts the impulses. So hopefully I'm in the right place. I'm a biologist, and I'm here actually today to present a body of work that's being done as part of a large NIH initiative. It's called the BioAssay Research Database, and we affectionately call it the BARD project. And I'm sure you can guess in a short while here where that term comes from. Thanks, Alex, for having us here. I think you've invited me to a lot of the user group meetings. I've never had a topic that means as much to me as this one does. And so we're here, hopefully, in a couple years, we'll be telling a story similar to what Sergey and Roland just shared with us, but with respect to the Assay Definition Standard. So, okay. So this is actually a very large cross-center initiative. It's part of the Molecular Libraries Program, which is a multi-million-dollar NIH initiative. And as we were all coming together during year 5 of this project, we realized that our ability to access and use all of our data out of PubChem wasn't easy. And so we had to come together and figure out what are we going to do. And when you work with the NIH or the government, one of the biggest challenges is can you be the impetus or the catalyst for the type of change that you need to have happen. So this group of scientists, if you will, are a living example that you can get the NIH to change. What we managed to do was actually convince the NIH that we wanted to get together and work collaboratively on a next generation bioassay research database. So this project is being led by the Broad and NCGC, and each of the other centers here each bring their specific domain or technical expertise to the table. So the University of New Mexico and Tutor-Prior, for example, bring their computational expertise. Vanderbilt is responsible and accountable for the outreach component, and it's as part of that that I'm actually here today. The University of Miami has lent all of their expertise with respect to the bioassay ontology, and Sanford Burnham and Scripps are actually key stakeholders who make sure that any solution we develop can be used by our internal stakeholders. So what are we here to do? We're actually here to try to facilitate or enable scientists, whether they're computational or more novice users, to use all of this valuable data that we've created. There are 7 of us trying to come together. It's a 2-year initiative. We've just completed year 1 of the initiative. As part of this project, we've also got some very specific grant aims that we must meet. We need to develop and adopt this Assay Definition Standard. After 23 years of working in the industry, I kind of feel a little bit embarrassed on behalf of the industry that we have the mole file, we have the SD file, we have SMILES, we have all different ways to represent structures, but we don't have a currency or medium of exchange for assay data. And in the age of public-private partnerships, it actually just makes good common sense that maybe it's time that we had an easy way in which we could exchange assay data and information. In order to get the scientists to adopt this standard, we also have to do certain other things. So we have to provide a repository where they can load all of their data. We have to make sure that we can enable them to do some predictive things that they'd like to do that they currently do in PubChem and other technologies. And we wanted to make sure that we played in the open source environment. So how I'm going to approach this talk, if you will, is kind of just tell the story through the lens of what we've needed to do in order to meet the grant objectives and aims. And if you can see that little yellow person in between all of the blue people there, this is my perspective. I think for those of you who've heard other talks, you probably get a very different view on this project depending upon which center was presenting the project. The Broad was the catalyst for the change. The project was originally termed the Project Phoenix. Some likened it to rising from the dead. You know, there were some inherent challenges with PubChem, but what you actually come to realize is that PubChem and BARD are going to coexist as part of an ecosystem to support the chemical biology community. I'm going to share what we've done with research data management and the Assay Definition Standard. I'm going to tell you a little bit about the technology stack. I'm not a technologist anymore. I've got a huge army of incredibly talented engineers that I think get along quite well with the ChemAxon engineers, and that's probably part of the reason that we've leveraged so much of your technology. And I'm going to share a little bit about the predictive models that have been implemented and set the stage for what we believe is the future for chemical biology research. So, a little bit of fun to keep you awake. When you look at that picture, what do you see right now? Anybody? What was that? Bark? See, now he's not paying attention. The Titanic? What do you see now? So this, if you will, I think is a great analogy of what happened with PubChem. If you looked at the surface, PubChem actually looks quite good. But then if you drill down deeper and you try to make sense or meaning of all of that valuable information, you can't. It's just impossible to some degree. So this is, if you will, our way of communicating what the value of context. And context is all of the annotations that are applied in a very systematic manner to all of that very rich biological data. So if I leave you with one message, it's that as you look to enter into this biology domain, the chemistry data type is complex, but with biology, the data is complex. And so it takes much more time and effort to actually address this very complex and vast field. So if I start you with PubChem, where we're headed is we want to actually structure that data. So look under the hood, and it's really only PubChem BioAssay that we're focusing on. How do you get to a point and place where you can actually see structured annotations? So we've created this structured data dictionary that significantly evolves from the current standard so that we can resolve some of the key issues that we had with PubChem. So examples would be, in PubChem, you really didn't have a true definition of what an assay was. You didn't have a definition of what an experiment was, and you didn't have the concept of a project. So we've resolved all of that with BARD. PubChem is very column header centric, so as we've been doing this data migration, this is probably the hairiest data migration I've ever been involved with. You've got thousands and thousands and thousands of column headers with all of the concentrations and the very useful experimental variable information embedded into those column headers. So we've actually transitioned to a result type and concentration name value pair model within the catalog of assay protocol that enables us to get a much better handle on that data. PubChem had a very extensive use of unstructured text. I'm not saying, or we're not saying, unstructured text is a bad thing. It's just that for the type of data that we're trying to handle, structured text is part of that equation. Long term, we plan to integrate both the capability to handle both structured and unstructured text, but we're transitioning here with BARD to a more consistent usage of language in order to facilitate meaningful interpretation of the data. We are not by any means replacing PubChem. There's a small component of PubChem BioAssay, that we think by restructuring the data and storing it in a better technical architecture will set the stage for hypothesis generation. So how have we done this? This has actually been led by a scientist. I strongly believe that the data steward for the data should be a scientist, so he's Dr. Josh Bitker. He heads up our screening group, automation and compound management group. Incredibly, incredibly bright scientist, and has coalesced a large team of scientists, industry academicians to come together and help us formulate what should our structured data look like. There's 4 things you have to define. This is the minimum amount of information that you have to define in order to define a new assay. You have to tell us a little bit about the assay. You have to focus a lot on what's the underlying biology. This is the most important part. The combination of these 2 things actually helps us define what is a unique assay. So we've got an algorithm that will then help us generate that assay identifier. You then have to tell us a set of information associated around what project, what therapeutic area, why are you doing this project? And then you have to give us some information about the results or the things that you're measuring. Underneath the hood here, how we're involving the current standard, if you will, is we are integrating with all of the public ontologies out there that make sense for us to integrate with. So we are heavily dependent upon the BioAssay ontology. We also integrate with gene ontology. We're picking up information from UniProt. So there's a lot of other information that we're helping the scientists integrate with without the scientists actually knowing the level to which they're helping us annotate their data. By trapping this information right up front, we're making it part of their day-to-day process so that all of those rich queries that they want us as technologists to be able to deliver back to them, we can then do that. So this sets the stage, if you will, for encapsulating all of this information in the Assay Definition Standard. So what does this Assay Definition Standard look like? I know Alex is heavily involved in the Pistoia Alliance, and we're hoping that we're going to be able to leverage his relationships there. This is a nascent standard. It's not fully fleshed out. We expect that it will evolve, and the more and more scientists that give us feedback and information on how it needs to evolve, the better our chances are of actually using this standard. So at the Broad, we have collaborations with several large pharmaceutical companies, and one of the biggest challenges I have as head of informatics is every time we do a new collaboration, then scientists want to exchange their data, we've got to come together and figure out how are we going to exchange that assay data. For structures, 2-3 weeks and we're done. We can figure that out quite straightforward. But assay data, sometimes it's months later and we can't convene on how are we going to exchange that data and information. So real quick, we're thinking XML or JSON. There's a set of information all surrounding the control vocabulary, this minimum information that we need. There's a set of information surrounding the experiment, points on the curve, replicates, activity, outcome score. There's a set of information around the results, and then there is room or ways in which you can add new information to the dictionary. So in the event that we don't currently have, or you're doing a novel assay, and we don't have the language or vocabulary that you can need, then you can go ahead and suggest that those terms be added, and the workflow components of the catalog of assay protocols will actually pick that up. So I'm a strong believer that when you work with scientists, you got to make sure that your technology stack actually mirrors what their process needs to look like, and you have to make sure that your entire value proposition to them is very clearly articulated. So our technology stack is listed here. You can see there's a pathway by which the novice users access the technology stack, and there's a process by which the expert users access that technology stack. It's a fairly straightforward workflow. The ability to define and register assays, the ability to deposit high quality data. Those annotations are really critical to doing all those types of queries that they want to be able to do. And they want to be able to do more Google-like searching. So we're, I would say quite politely, tired of forms. It's not that we didn't love ISIS. It's just for those of us who've adjusted to Google and Amazon, we want to see, can we actually take some of the technologies and methodologies that the rest of the world are using and apply them to our data. So that's where we're headed, is real easy to use interfaces. So this translates into 3 different sets of things that we have to accomplish. The first is the catalog of assay protocols. That's a tool that's going to be open source. That's your registry, if you will, of all of your novel assays. The middle section here, it's a Spotfire dashboard. The most difficult part of this project is actually migrating all of that PubChem data. We've got about 5,000 to 6,000 assays that we need to migrate. But after we take or extract all that information out of PubChem, we literally have to manually annotate it to a certain standard and then curate it over time. And then there's a quick snapshot of the 2 different client applications we've got. One is actually the BARD web portal, which will be released to a limited audience shortly here. And then there's a desktop client for the more computationally savvy users. So what does the BARD web query tool look like? So as I mentioned, Google-like searching, I can go on there and just look for things like DNA repair. There will be a body of data that will be added, compounds, assays, and projects. We've got an Amazon-like query cart, so as you walk through here, you can click on things that you'd actually like to save to that cart for future analysis. Right here, there's filters. And depending upon as you navigate from assay to compound to projects, your filters will change. And these filters, the reason we're able to give them the ability to search by all of the filters and the facets here is largely a consequence of actually annotating that data. So we're using a very rich vocabulary and language, if you will. So here's an example project view. You know, I think I heard someone today mention Lipitor. But the next generation of products that we're looking for is for those subset of patients that actually don't respond to Lipitor. So here's a really interesting project. It's a scavenger receptor ligand binding where we're looking to actually increase the quantity of HDL. So you can see some of the annotations are around the project-specific items. You can see here that you've got some of the annotations around the biology. And you can actually click on these links and go out to UniProt, go out to all of the other entree sources to navigate, if you will, across all of the NIH sources that have come to being over time. Another example here, you know, for years, we've been doing screening campaigns. So one of the views we've developed is actually a view where they can traverse the screening campaign. So you start with your primary, do your cross assay analysis, and you keep going and doing your rounds of lead up, et cetera, until you get to your probe. Viewing all of this, you can click and link out to other information, or you can continue to do it, if you will, in your more traditional SAR type worksheet. We've got actually the linking to the curves. We're only representing the images of the curves, replotting them using the parameters that have been input during the analysis. We're not actually recalculating information. Other examples of visualizations, we're time constrained here. So here's an example of a sunburst visualization. And we're actually using the target classifications from Panther here. But green basically means that you've got high activity against a subset of classes, and red, in this case, means you don't have sufficient activity. So over time, there's a library of visualizations that we're adding, and we've actually got a very strong user community that we do usability studies on the web UI and make sure that they're consistently giving us feedback on what should and shouldn't be included. We have an internal advisory panel that makes sure that we're meeting the grant aims and are delivering something that's of value to the community. And then we have an external scientific advisory board that includes a combination of fairly senior leaders from both industry and from academia who kind of give us the no-go decisions that enables our project science officer to decide, yes, you will get your next round of funding. So real quick on the technology. So you know, we do have a data warehouse behind the scenes. So the data from the catalog of assay protocols gets extracted out into the data warehouse. And then the web and query desktop clients actually access all of that information. So, you know, we're using a mixture of internally developed, and where it's necessary, we link out and use other things. So we are using D3 for the data visualizations. A lot of the searching has been enabled through Solar. I think Sergey alluded to this when he talked about collaboration. So this actually is a good example of you know, the expertise across the different sites coming together. I think one of the engineers in my team actually prototyped some of the work that we needed to do on text searching. But then when we looked at the technology stack, it made sense for that to actually belong behind the REST API. So he transitioned that over to NCGC, who's actually implemented it in the right, or at the right level within the technology stack. ChemAxon, you know, whenever you have a project with 7 different centers and 7 different cultures coming together, there's going to be some things you have in common and some things you don't have in common. ChemAxon would be happy to know that ChemAxon is something that we just generally agree on. So how are we using ChemAxon and BARD? We're using it in the REST API and the data warehouse. We're using it for rendering structures. And actually for our promiscuity plugin, this is the Bad Apple plugin that I'm sure some of you are familiar with, that just gives you a quick estimate of how promiscuous your compound is likely to be. The University of New Mexico are heavy believers in the ChemAxon technology stack. In the desktop client, so it's being used for rendering structures, molecule import and export, and Marvin for drawing query structures. So you'll see it throughout the query and analysis tools. Predictive models, so what we're hoping with these predictive models, I lost count of the number of different algorithms and methods I've heard in the last 2 days, but one of my lessons learned from working and supporting scientists for so long is we're really good at developing the algorithms. I don't feel we exploit and use them as much as we could or should. So what we wanted to do when we envisioned this project was actually create the technical infrastructure so that you could have a very easy and seamless way by which you could deploy more and more algorithms. So we're actually going to go live with 1 or 2, maybe 3. And then the hope is that the community will actually engage with us and provide us algorithms that can then be applied to all of our different data sets. So the first example of this is actually the bad apple algorithm. And what it does real quickly is it just scores, 0 to 100, the compound's not really promiscuous, 100 to 300, maybe some activity. And then above 100, definitely a promiscuous compound. Another example, this one actually is Patrick Rydberg, and I think it's Lars Olsen out of Denmark. And there's 2 views, but it basically predicts CYP450 isoform metabolism sites. And it actually uses a 2D fingerprint to do that. There's a summary view and then a data view that just real quickly gives the scientists some guidance around what the intuition for what the compound of the probe is likely to do in a scenario like this. So, back to where we started, I think one of the young women here, mentioned that she thought I was her age, and I took that as the ultimate compliment because I think I was probably old enough to be her mom. We've been doing this for a really, really long time, and being on the customer end, one of the things that's quite difficult is to navigate this maze. You've got so many different vendors, you've got so many different tools. How do you pick the right tools and make sure you're not internally developing what ChemAxon is developing? And so if there's one wish I would have for Alex and Chizzy, is I feel like you do have a corporate strategy, but it's not completely clear to me. And if I knew more about your corporate strategy, I could potentially make different or better decisions on the timing of some of my projects and what I'm going to opt to internally develop so our timing better aligns. So that's one of my lessons learned from the last 2 or 3 days. They're also embedded in here. You'll see a whole host of very domain-specific tools, but once in a while you come across a tool that I believe actually is a good example of something that our industry ought to emulate. So as you're thinking of your next generation platform, especially given your strong engineering culture, I would highly recommend that you take a look at MicroStrategy, because as I was trying to put all of the puzzles, pieces that have been given to us over the last couple of days, I think you've got an incredibly unique opportunity ahead of you to become the market leader in this space. The first time I think I looked at ChemAxon, I worked at Cytokinetics, and back then Daylight was the company to beat, and ChemAxon was just entering the market. So it's 12 or 15 years later, and I think ChemAxon's the company to beat. And unfortunately, I don't even know if anybody actually still uses Daylight. So that's kind of like my position on where we are in the marketplace. So where are we headed, and what can we do together? Where we're headed is we see BARD as the open source platform for translational research to support NCATs and all of the next generation chemical biology initiatives. When I look at what we're trying to do, yes, we are going to provide tools, but I think one of the most valuable things we want to try and do is create and set the stage, if you will, for implementing this Assay Definition Standard. In the age of public-private partnerships, we heard Lily talk about open source innovation. We're seeing a lot more of us come together in ways we wouldn't have envisioned 10 or 15 years ago. This becomes more and more important just so we can exchange data and information in an easy way. I think the tools will continue to evolve. That's just the nature of what we do, whether it was hearing from GSK, who talked about the transition from ISIS HViews into IJC and potentially into something else later on. Our tools are constantly evolving. I fully expect that to continue. So what ends up being real important is the data sets. What are these valuable data sets, and what are we going to do with them? So we're starting small. We've got our molecular libraries program, 5,000 to 6,000 assays. We're going to go after the NCI 60. We'll probably take a subset of chemical data. Tutor has some strong relationships with OpenFacts. We've got other parts of our center that have good relationships with St. Jude's Medical Research Center. We've got some pharmaceutical companies who have actually open sourced some of their data for malaria, TB. So we imagine that we'll slowly prioritize data sets that get added to this resource. Methods, talked about at Bad Apple, talked about SIP 450. We're hoping the community will engage with us here. And my vision here is, if you will, is there's a lot of knowledge that the expert users have when they create these algorithms, but that those algorithms and the logic and the meaning of that is quite difficult to transfer to the less expert users. So what the vision is to try and get these algorithms proceduralized to the point that a naive user can more easily interpret and use them. So exploit the algorithms as much as you can. Build once, reuse over and over again. And then last but not least, we're hoping to get to the point where you're using KNIME and Pipeline Pilot to do a lot of workflow. So I think Bark wants to come see a tool that we recently published that actually encapsulates our best practice for hit calling and cherry picking into some Spotfire guides with our diversity-oriented synthesis libraries, which was quite tough to develop because you had to get the scientists to agree on what their process was, and then you had to implement the workflow, and then you had to deal with some technology challenges. So that last box there, if you will, is all of this is about analyzing the data and making sense of it. What we're here to talk to ChemAxon in particular about is we don't currently, as an industry, have a best practice for an open OLAP model. If you compare what industry and academia has been leaning towards over the last couple of years, we all seem to be convening around some sort of dimensional model. So through Doug, who we've known through years, and Alex, we're in conversations to ask, is there a way in which, through the combination of this Assay Definition Standard, can we set the stage for just some sort of data model that we all agree on? And if we then have that combination of both of those for small molecule science, are we making a significant leap forward in terms of filling in some of these gaps that we haven't managed to address in the last decade or so? So that's where I was going to end.
No. Right now, it looks like it's dose response and single point data kind of centric. Are you thinking about MetStab and PK? And
so it will do MetStab because Vanderbilt actually does all of our ADME PK data. And one of the things we did was when we defined the assay catalog and the language and vocabulary is we made sure we could handle their assays.
And PK?
So PK, I think it will do PK too.
So
what
you've
I mean,
got vehicle and stuff like that.
It should theoretically, yeah.
You
so
think you can actually?
Yeah, so what we did here is, coming from industry, this was my first nonprofit assignment, and it was actually frankly shocking to see the state of the data. It just didn't occur to me that the data could be in as bad shape as it could be. So then I tapped all of the industry veterans that I worked with, including a data architect who worked at Amgen. And so Amgen has been part of this, Novartis has been part of this, and so they're all used to handling data across the entire value chain. So we've intentionally tried to build the underlying data structures to actually handle some more of that complex data.
Contact Gilson. He was at NIST and he did the standard for the calorimetry and assay data. So there was already a standard out there.
So I don't know Gilson. So that seems like a name that I need to know.
Heather Carlson, she has her own database for capturing assay data as well. There's several of them out there.
Yes. So I don't know that one. So can you email me that one? Yep.
There's 6,790,000 Campbell assays out there, and they're individually curated on outsourced basis. How are you going to handle those?
I don't think we're going to handle all the Campbell assays. So we have a committee that helps us prioritize what's going to get in there. And I think the Campbell assays, they manually curate them, right? They extract the information out of papers, the one-off type stuff. So I don't know necessarily that we're going to go for that type of information. I'm,
You add up all the one-off, it's more than the library screening industry.
Yep.
Compound based.
yep.